The third and final presenter in this morning's session is Dr. Josh Mendell. He's a Howard Hughes investigator. He started his career at Hopkins and recently has moved to UT Southwestern, where he's professor of medicine molecular biology program there. Dr. Mendell.
My laboratory has been interested in non-coding RNAs for over the last decade, and we've, for the most part, focused most of our attention on the microRNA pathway, which we haven't heard about today. And we've tried to understand how these small regulatory RNAs are themselves regulated and what functions they perform in physiology. I decided today to talk about, more in line with some of the other talks we've heard about today, the functions of this much more enigmatic class and much more diverse class of RNAs, these so-called link RNAs that we've already heard so much about today. And what I'll try to do, at least early in my talk, is sort of discuss how some of the approaches that we've taken to understand small RNA biology in mammalian systems we're now applying to try to understand some functions of particular link RNAs. Now, we've heard a lot about the functions of link RNAs in the nucleus already today and how they can regulate transcription both in cis and trans. But this is really just one aspect of the functions of these very diverse RNAs, and I'll try to give a flavor for that today with a particular example that we're very excited about. So these RNAs, some of them leave the nucleus and go out into the cytoplasm, as we heard earlier from Lynn, and interact with either target RNAs or proteins and regulate their activity. And so this is a very distinct type of function for link RNAs than what we've heard earlier today. And I'm going to provide an example of that. So I mentioned that we've been very interested in the microRNA pathway, and we've been particularly interested in how these small RNAs play a role in disease, and in cancer in particular. And one of the ways that we've approached the problem of identifying microRNAs that participate in both normal physiology and disease has been to ask how they participate in broader signaling and transcription factor networks that we already know are very important in these processes. And that's turned out to be a very fruitful approach for us, and we've looked at some of the most important pathways in cancer, for example, like the MYC pathway, the KRAS signaling pathway, the P53 transcription factor network, and we've found that microRNAs really participate and play important roles in these networks. And so we've wondered more recently, with the emergence of this new understanding that there's this very diverse class of link RNAs, we've wondered whether this type of non-coding RNA has similarly been functionally integrated into these core signaling networks. And the work that I'm going to talk about today is largely the work of a graduate student in my lab, Sung-Yul Lee, who is defending his thesis next month, and with some very important assistance from a relatively new postdoc in my lab, Florian Kopp, as well as a technician and faculty member in my lab, Anu and Sung Chang. Now, this work really started in my lab with a screen to try to identify microRNA or link RNAs that might participate in the P53 pathway and in DNA damage signaling. And because I have limited time and I want to get into the details of this link RNA, I'm not going to go into details of that screen, but we came across an RNA in the process of those experiments that we found was induced by DNA damage, in this case by treating cells with the DNA damaging agent doxorubicin in a P53 dependent manner. And I admit that the induction by DNA damage is somewhat modest, and at first we weren't that excited about really the magnitude of increase after DNA damage. It's less than twofold. And if there's time at the end, I'll come back and tell you why we now think that that amount of induction could actually be very functionally relevant. But what this really led us to do was to take a really close look at the RNA. And that's when we really got interested. And what we found is that this RNA is an approximately 5 kilobase transcript that is very highly represented in publicly available RNA sequencing data sets. And it's very conserved. Unlike a lot of link RNAs like we heard earlier, this one is very conserved over this 5 kb region, and with 65% nucleotide identity between human and mouse, which is quite high for a lincRNA, as we've heard. And we also looked at its expression in other publicly available sequencing data sets, and it appeared to be ubiquitously and abundantly expressed. And we could confirm that by simply performing northern blots in some cell lines. I'm just showing this one particular cell line that we like called HCT116, which is a cell line that's been widely used to study the DNA damage response in the cell cycle. And it's expressed as a discrete transcript of the expected size. And remarkably, when we actually measure the copies of this transcript per cell using a quantitative PCR assay, we see that it's present in mammalian cell lines at approximately 500 to 1,000 copies per cell. And so this is in the range of an abundant messenger RNA like beta-actin, which is also thought to be about average of 1,000 copies per cell. So this is a very abundant, very conserved RNA that's ubiquitously expressed, which seemed to distinguish it from a lot of the lincRNAs that were under study. And that's really what attracted our attention to it. Now, we convinced ourselves that this really was a non-coding RNA as opposed to a protein-coding gene by a variety of metrics, but one of the best ways to distinguish protein coding from non-coding genes is to look at their evolutionary signature. So protein coding genes, as they evolve, accumulate substitutions that are biased towards the wobble position, or when amino acids are substituted, conservative amino acids are substituted in. Whereas non-coding RNAs are not subject to those constraints. And there are various metrics that you can use to actually measure that. And when you apply one of those metrics called phylo-CSF to this transcript, which I should mention we've been calling non-coding RNA activated by DNA damage, or NORAD. When you apply this metric to NORAD, you see that there's really no detectable conserved open reading frame anywhere in this transcript. And the highest scoring open reading frame scores similarly to other known lincRNAs such as exist, that we've already heard a lot about. So as far as we can tell, there's no conserved open reading frame, and the conservation of this transcript is not being driven by a protein coding function. So what is it? What does it do? And so to really start to get at what this RNA might do, we decided to use a loss-of-function approach. And we took advantage of these recently developed genome editing technologies. In this case, we used tail-end nucleases, which are custom nucleases that we could design that would allow us to precisely and efficiently insert a transcriptional stop element into the very beginning of the transcript. And we designed multiple tail-ends and generated multiple cell lines lacking expression of this NORAD transcript, as you can see. And this turns out to be a very efficient method to knock out the lincRNA. So at first, we took a look at the known DNA damage response in these cells, which is a very well-characterized G1 and G2 cell cycle arrest. And we were a bit disappointed to find that those checkpoints were completely normal as far as we could tell. But we noticed something quite interesting in the course of those experiments. And that is, when you analyze the cell cycle, you look at DNA content by staining cells with a DNA staining dye. And of course, this cell line, HCT116, is a stably diploid cell line, so they have 2N and 4N DNA content. Now, because we generated a large number of knockout clones, we looked at the cell cycle in a large number of these independent knockout clones, and we found that a significant subfraction of these knockout clones were actually had tetraploid DNA content, at least according to this flow cytometry assay, meaning they had twice as much DNA as the diploid parental cells or most of the other knockout cells. If you actually look at the chromosomes in these cells, this cell line, as I said, is known to be a chromosomally stable diploid cell line with 45 chromosomes. Most of these cells lack the Y, which is why there's 45 chromosomes. And if you look at these tetraploid, apparently tetraploid cells, you see that really there were actually two things that we noticed. First of all, I don't know if you can see these numbers, but this is the number of chromosomes in the cell. And there are approximately twice the number of chromosomes as you would expect. But the other thing that you might notice here is that now the number of chromosomes is actually variable between the cells. And this suggests that these cells may have adopted a phenotype called chromosomal instability, or SIN, which really refers to a numerical instability of chromosomes. And so that led us to wonder whether the cells that were actually diploid by the flow cytometry assay might also have now variable numbers of chromosomes. And indeed, when we looked at them through this low throughput assay, where we're just looking now at metaphase cells, we could see that each metaphase had roughly a different number of chromosomes. Now this is a relatively low throughput way of assessing this type of phenotype, because we really have to hunt around and look for these metaphase cells. And so we can assess this phenotype using a more quantitative method that's been widely used to study this phenotype, which we call chromosomal instability, or CIN. And the assay is based on fluorescent in situ hybridization, and I'll mention in a minute. But the reason it was really developed is because this phenotype has been studied for over 100 years and and is now recognized as a hallmark of cancer cells. And so there's been a lot of interest in understanding what mechanisms control the numerical chromosomal stability of the genome and how that goes awry in cancer. And the role of lincRNAs in this process is very poorly understood. And so we were excited by the possibility that this lincRNA might be regulating this process. So the specific assay that I alluded to is this FISH assay. It's probably hard to see, but here we're just essentially taking some marker chromosomes and labeling their centromeres in interface cells, and then we can count the number of cells that have normal numbers or abnormal numbers of these marker chromosomes. And so in wild-type cells, every cell, for the most part, has 2 reds and 2 greens, 2 chromosome 7s, 2 chromosome 20s, whereas NORAD knockout cells frequently exhibit chromosome loss or gain. Here's a cell that's missing chromosome 20, for example. And you can now count this in hundreds of cells, and you can score the number of cells that have a non-diploid content for these particular marker chromosomes. And when we do this, we see that there's really a great elevation of the frequency of aneuploidy in these knockout cells. And so these are 3 different knockout clones generated with different tail-end nucleases. And remember, we're only assaying 2 chromosomes in this assay. So this is probably, even though there's about 25% of cells now that are aneuploid, there's probably many, many more cells that are aneuploid if you looked at all the chromosomes, which is more in line with what we saw when we looked at the metaphase spreads. So loss of this lincRNA is causing a dramatic increase in aneuploidy. Now, we believe this is due to the primary generation of aneuploid cells, as opposed to another model where you might imagine that these cells are just more tolerant of aneuploidy. And so the cells might accumulate in the population because they would otherwise die. But when we actually image these cells using time-lapse microscopy, and we did this in collaboration with Hong Tao Yu's lab and a very talented postdoc, Sushama Sivakumar, we could actually see that there's a very high rate of mitotic errors in these cells. If you image them over time as they go through mitosis, we can very frequently see anaphase bridges and mitotic slippage. Mitotic slippage is when cells enter metaphase but don't actually go through anaphase. And that probably is what produces the tetraploid cells. So the cells really just don't complete mitosis. And so we can see these errors in these cells in live imaging, which means that we're really producing de novo abnormalities in the genome content when you lose this lincRNA. So I'll go through this quickly, but I want to mention that we were very careful early in these studies to establish that the phenotype that we're seeing is really due to loss of the lincRNA, and not instead the possibility that this would be an artifact of genome editing in these cells. Because remember, we generated these knockout cells by creating these synthetic tail-end nucleases and introducing them into cells. And you can imagine that there's the possibility that that could lead to DNA damage and itself cause aneuploidy. But we've done a number of experiments to really rule that out. And we don't really see any evidence that these genome editing technologies cause this phenotype. And I'll just very briefly run through that so I can get to the more interesting mechanistic work. But simply, the first thing we did was just get use a different published tail-end that targets a totally unrelated locus. And we targeted cells with these tail-ends, and we never observed any tetraploid subclones after targeting with this. And these cells are not aneuploid. We also used a completely unrelated method, short interfering RNA knockdown of NORAD, Here, we can only really knock down the transcript for about a week at a time when we transfect them with siRNAs. But remarkably, that's actually sufficient, even in that period of time, to result in a measurable increase in aneuploidy. And so this is a totally unrelated method of inhibition that has nothing to do with genome editing. And what's even probably more convincing even than this is that if we then allow the siRNA to wear off and then subclone the cells after that, we now see for the first time de novo generation of tetraploid subclones only from the siRNA transfected cells. And so finally, the last thing we did is, because of the way we generated these knockout clones, we integrated these transcriptional stop cassettes into the endogenous gene, but the transcriptional stop cassette is flanked by LOXP sites. So we can actually remove it and revert the cells. And when we do that by expressing CRE, we can turn NORAD back on, and then we can look at what happens in these cells, and we see that when we derive subclones from these compared to control-treated cells, the rescued cells have much lower levels of aneuploidy. So all of these experiments really convinced us that we were studying an on-target effect. In other words, this link RNA was really controlling this phenotype. Now, the last sort of, I wouldn't call it a control experiment, but an important experiment was we wanted to show that this was not a feature just of this particular cell line that we had done all the experiments up to this point in, because it is a cancer cell line, and it is transformed, even though it's stably diploid. And so we also targeted NORAD in TERT-immortalized primary human fibroblasts, which are non-transformed cells, and we see a similar phenotype. So all of these things convinced us that we were onto an interesting RNA that controls an important phenotype. And of course, now the million-dollar question always is, what is the mechanism? How does it do this? And I'll just go back to this scheme one more time. One useful initial discriminating feature of link RNAs that might help give you a clue of its function is simply to look at its subcellular localization. And when we did that with NORAD, we were actually surprised to see, although you probably can't see, that NORAD is actually almost exclusively cytoplasmic. You can see it over here. This is a qRT-PCR fractionation experiment. And it's almost entirely detectable in the cytoplasm, as much as a beta-actin messenger RNA. NEAT1 is a known nuclear link RNA. So, and we see the same thing by single molecule RNA fish. So this RNA is completely out in the cytoplasm, which was a bit of a surprise since it seems to be regulating the genome, but a very important clue. Now the second important clue was that when we looked carefully at the NORAD sequence, we noticed that there are regions within the sequence that actually repeat, sequences that repeat. And you can see this by aligning NORAD to itself using the BLAST algorithm. So this is simply the NORAD sequence aligned to the NORAD sequence with each dot representing identity. And so, of course, there's a 1-to-1, but then there's also these offset regions of nucleotide identity. So region 1000 is also identical to region 3000, for example. And you can deconvolute this, and what it actually looks like is there is a domain, which we were calling the NORAD domain, which is repeated 5 times in the transcript. It's about 400 nucleotides long. And you can see that most of the conservation of the transcript is centered over these domains, although there is some additional conservation over here. And we hypothesized that these domains are nucleating a ribonucleoprotein complex in the cytoplasm that would, this domain would represent a binding site for some key component of that. And so what we did is we broke the transcript into these pieces, and we synthesized these fragments and corresponding antisense controls, and we did pull-down experiments in mass spec identification of interacting proteins. And we looked for things that would bind to each and every one of these domains and none of the controls. And using that criteria, actually only a single protein came out of this experiment, and that was a protein called PUMILIO2. And this is just a representation of the mass spec data, but you can see that if you pull down these RNA fragments with these NORAD domains, they're also in the 5' end of NORAD as well, we can, by Western blot, very efficiently recover this protein PUMILIO2. And we can do the reciprocal experiment. We can pull down PUMILIO2. There's also a highly related protein called PUMILIO1. If we pull these down from human cells and then look at NORAD association, we can see very, very significant enrichment of NORAD in these immunoprecipitates, over 1,000-fold enrichment. So it seems to be binding very, very well. So what is PUMILIO2? So PUMILIO1 and PUMILIO2 are members of a very deeply conserved family of RNA binding proteins that act as negative regulators of translation and mRNA stability. They bind to sites typically in the 3' UTRs of target messages, and they trigger deadenylation and, as I said, inhibit translation. And one distinguishing feature of PUMILIO proteins is that they're exquisitely specific. Unlike a lot of RNA binding proteins that have more flexibility in terms of the sequences they can bind, PUMILIOs are very, very specific for this 7-nucleotide recognition element shown here. And so we can look for that element in NORAD, and we expect to see that element occurring approximately once every 16 kilobases by chance. And remarkably, we can find, if we only allow a perfect match to this motif, we can actually see 15 very highly conserved occurrences of these PUMILIO response elements, or PREs. And you can see that most of them fall within these NORAD domains. And this is by far the most PREs of any transcript in the transcriptome. Most targets have 1 or 2. 90% of targets have 2 or fewer. So this is really an outstanding example of a potential target for PUMILIO. Now, up until this point, all the data I've shown you regarding the interaction of PUMILIO with NORAD has been through binding experiments where we break open cells and pull down NORAD or pull down PUMILIO. And the problem with those kinds of experiments, while useful for screening, is that it's known that when you break open cells, RNAs and proteins can reassociate. And we've already heard today about how important it is to do experiments involving UV cross-linking to really capture endogenous interactions in whole cells. And fortunately, in part because PUMILIO is so specific, a CLIP experiment, a CLIP-seq experiment, which involves cross-linking the cells and then identifying all the binding proteins, has already been performed for PUMILIO2 by Tom Tuschl's lab. And they identified 7,500 binding sites for PUMILIO2 genome-wide. And remarkably, in their data, we were really excited to see that NORAD, a site in NORAD, is actually the 11th most abundantly represented CLIP cluster in their entire data set. So we were really excited by this, but then we realized something. When we were looking at the NORAD sequence, we noticed that there are actually a lot of sequences scattered around the human genome and the mouse genome that are very similar to NORAD. There's dozens of these sequences, actually. And a few of these sequences are almost full-length copies of NORAD. But we've looked very carefully, and we can't detect expression of any of these. And if you look at them, they have features of pseudogenes. Processed pseudogenes are often products of retrotransposition. When they go into the genome, they create a target site duplication, and there's a poly A site that's in the genome, encoded because they were reverse transcribed. And you can see many of those features for most of these. So these most likely represent pseudogenes that are not expressed. So functionally, we don't necessarily think they have any importance, although it's possible in some settings they might. But the most relevant thing about this is that when one does these high-throughput sequencing experiments and maps the reads back to the genome, a typical filter is to only analyze reads that map to a single place in the genome. And when we noticed this, we realized that in the data set produced by the Tuschl lab, they actually probably disposed of most of the reads that map to NORAD because of this feature. So we reanalyzed their CLIP data, taking into account this feature, and remarkably, NORAD was far and away the most highly clipped gene in their entire data set. And so, I should mention that they did this experiment using epitope-tagged PUMILIO2. And so we've now done a CLIP experiment with endogenous PUMILIO2 in this HCT116 cell line that we've been working in. And our data, I will say, are not as deep in terms of target identification as the Tuschl data set, in part because the antibodies for the endogenous PUMILIO2 are not as clean as the epitope antibodies. But nevertheless, we see a very similar pattern with endogenous PUMILIO, and NORAD is far and away the most highly clipped target. So based on all of these data, we hypothesized that NORAD represents a very strong binding platform from PUMILIO in the cytoplasm. It's present at approximately 1,000 copies per cell. It has probably at least 15 binding sites for PUMILIO. So it can sequester up to 15,000 molecules of PUMILIO per cell. And when NORAD is deleted, this pool of PUMILIO is then released to hyperactively repress targets. And so this model really predicts 4 things that we've tested, and I'll go through these experiments quickly. First, in order for this model to be true, there has to be favorable stoichiometry between NORAD and PUMILIO. If there's 1 million molecules of PUMILIO in the cell, 15,000 binding sites is not going to make a difference, obviously. Now, assuming that we can satisfy that criteria, we would expect to see that evidence for PUMILIO hyperactivity in NORAD knockout cells, and that can be seen by looking at the targets of PUMILIO and whether they're repressed. We might also expect to see that overexpression of PUMILIO can phenocopy NORAD loss of function, whereas knockout of PUMILIO might be able to rescue it. So we've tested each of these predictions, and first I'll show you the stoichiometry. We quantitatively measured the number of PUMILIO protein molecules per cell. Remember that there's about 7,500 to 15,000 binding sites for PUMILIO provided by NORAD. And it turns out that there's actually about 15,000 PUMILIO protein molecules per cell if you combine the number of PUMILIO-1 and PUMILIO-2 molecules. So we've done quantitative Western blotting using standard curves here. And so there's under some conditions, there's a 1-to-1 ratio or a 0.5 to a 1-to-1 ratio. So the stoichiometry is very favorable for this to be performing a sequestering function. So next, we looked at the PUMILIO targets in NORAD knockout cells using RNA-seq. And we could see statistically significant repression of that set of targets, which is consistent with PUMILIO hyperactivity. We also then overexpressed PUMILIO-1 or PUMILIO-2, and we see that the genes that are downregulated in NORAD knockout cells are similarly downregulated when we overexpress these proteins. And we also see that when we overexpress these proteins, PUMILIO-1 or PUMILIO, and in particular PUMILIO-2, that's sufficient to induce measurable aneuploidy as measured by this chromosome fish assay that I showed you earlier. So finally, the last prediction is that knockout of PUMILIO should rescue the phenotype of NORAD loss of function. And so this experiment is a little tricky because there's 2 PUMILIO genes and NORAD, so that requires knocking out 6 alleles to really test this. But we've done this now, and we've taken advantage of the CRISPR vectors that our speaker this afternoon is going to talk about, Feng Zhang, to knock out PUMILIO-1 or PUMILIO-2 and individually first. And when you knock those out, we see a measurable decrease in the level of aneuploidy when you knock out NORAD, but it's not a rescue. And that's probably because PUMILIO-1 and 2 are redundant. And so next we made the double knockout of PUMILIO-1 and 2. And it turns out that this is actually harder than we expected. It was very easy to make PUMILIO-1 or PUMILIO-2 knockout cells, but the frequency of generation of the double knockouts was very rare. We were able to derive clones, which were useful for this experiment. But it turns out that knocking out, cells do not like to have no PUMILIO. If you knock out both alleles of both proteins, both genes, it actually is sufficient to induce a measurable level of aneuploidy in these cells. And so what this is a very interesting result actually, and I think it's relevant to the broader function of this non-coding RNA. So I showed you earlier that overexpression of PUMILIO causes aneuploidy. And we also see that too little PUMILIO causes aneuploidy, suggesting that there must be a mechanism to buffer the activity of PUMILIO in a very, very narrow range, which is actually what we think NORAD does. But this was still a useful experiment because we were able to then further knock out NORAD in these clones. And although the baseline level of aneuploidy is higher than background, there's no additional increase when you knock out NORAD. So I think these data are very convincingly show that the function of NORAD is mediated through PUMILIO. And we did one last experiment that further showed this. Because we have this time-lapse assay working with Hongtao Yu's lab, we could then transiently knock down PUMILIO and see if we see a reduced frequency of mitotic errors. And so we knocked down PUMILIO-1 and 2 together with 2 different pools of siRNAs, and in fact, that's what we see, a greatly reduced frequency of mitotic errors. So all of these pieces of data really point to this model. Now, so I think we've established that NORAD is a strong negative regulator of PUMILIO activity, and PUMILIO is important to suppress this chromosomal instability phenotype. But the question is, why does PUMILIO hyperactivity result in aneuploidy? And these proteins, although they've been deeply studied, they're deeply conserved all the way to yeast, actually. They've never been implicated in cell cycle control or especially genome stability. But when we looked at the targets of PUMILIO that are altered in their expression in NORAD knockout cells, we found there was really a remarkable set of genes that are highly enriched for regulators of the cell cycle and DNA repair and mitosis. And if you look at individual genes in this group, many of these genes have individually been shown to be critical for maintaining chromosomal stability, including core components of the cohesin complex, centromere components, and factors necessary for DNA repair or replication. And these genes, as you can see, are coordinately downregulated by this hyperactive PUMILIO activity. And we think that's why when PUMILIO is hyperactive, and probably why when PUMILIO is hypoactive, you would have a coordinated upregulation of many factors and really dysregulate the mitotic program in that way. And that's why we think PUMILIO levels have to be maintained in this very narrow range. So let me just summarize very briefly by mentioning that we think NORAD is this regulator of PUMILIO that maintains its activity in this very narrow range. When this is perturbed, PUMILIO can hyperactively repress a program of mitotic regulators and DNA repair factors and DNA replication factors whose dysregulation is known to cause chromosomal instability. And one of the interesting, I think, lessons from this work, in addition to how a non-coding RNA can control these PUMILIO proteins, is really how critical the dosage of PUMILIO proteins are in mammalian cell biology. And there was some hint of this earlier this year from Huda Zogby, who showed that knocking out a single allele of one of the PUMILIO genes, PUMILIO1, leads to neurodegeneration in mice. And we've similarly found, as I showed you, that loss of PUMILIO activity is deleterious in these cell lines, as is hyperactivity of PUMILIO. So it appears that this NORAD-PUMILIO axis emerged probably to really act as a very strong buffer to keep the amount of free PUMILIO within a narrow range. So let me just end. I've already mentioned the people in my lab that did this work, in particular Sung Yul and Flo. And we have some really important collaborators on this work. Hong Tao Yu, I mentioned, we did all our live cell imaging work in collaboration with his lab, but I need to also mention the very important role played by Yang Zi and Beibei Chen, who are very excellent bioinformatics collaborators and played a really important role in these studies. And I'm happy to take questions.
We have time for a couple of questions.
Josh, beautiful story. So are there any mutations then in NORAD or PUMILIO in cancers that have SYN?
Well, we've looked in some TCGA data, and I would say that we haven't done this. I wouldn't say we've completely comprehensively done this, but we have taken a first look. We haven't seen any evidence of deletions of NORAD. I wouldn't expect point mutations because I think there's a lot of intramolecule redundancy here with all these PREs. But we have seen in some tumor types evidence for amplification, although I will say that these are very big amplifications, so it isn't at all clear whether NORAD is the target of that. And at first that was sort of puzzling, but I think now that we know that too much or too little PUMILIO can cause SYN, that might actually be relevant. And I think now, based on what we've found, it's really important actually to look at PUMILIO as well, which we're really just doing right now, to see whether it is mutated or amplified or deleted, because that may be the more proximal regulator.
So I'm curious how you're thinking about this. Why would cells want to use NORAD to regulate RNA stability instead of regulating PUMILIO itself or themselves? And do you see large changes in NORA expression depending on physiological states?
So great question. And obviously I can only speculate. But this goes back to our initial sort of observation that NORAD is induced after DNA damage. And at first we were not all that excited because it's, if it's less than twofold, that's like the mantra. Just throw it out. It's not important, right? But if you think about it, if twofold change in NORAD actually produces a huge number of PUMILIO binding sites, so that can have a profound effect on PUMILIO availability. So we're really starting to look at how that regulation occurs now and its consequences on DNA repair and DNA damage, especially now knowing that PUMILIO regulates a lot of these targets that are involved in that process. So I do think that there's got to be dynamic regulation. I mean, we can certainly see it under this one particular situation. And we're looking at other stresses to see how broad that is. I think that that must be the answer as to why this system has evolved. It may also be that it's a convenient way to sort of provide a buffer, because regulating the amount of PUMILIO up or down may not be temporally precise, or it might not be tight enough control. Whereas if you have this buffer out there, it almost doesn't matter how much, if there are small fluctuations in PUMILIO levels, that should be buffered by NORAD. And so maybe that's part of the answer as well.